So thanks to Deepa and Kavita for actually inviting me here. Um, so the first picture was actually in our field station in Mudumalai where I was doing my master's. So I put that up simply to get you excited, but obviously I'm not going to be talking about elephants today. But I'm hoping that now that I'm back in India, I will be starting to do some going back to elephant research. So today, this talk is going to be kind of like quite different. So I was telling, I don't remember who, but I was saying that I'm feeling really very insecure when these people are talking about these experimental evolutionary scenarios where they're saying that we only have 10,000 generations and only like 5 million like individuals and stuff like that. So we worked very hard. We have only 1,000 individuals and we only have 30 populations. So obviously when we are working with natural populations, we have a lot of noisy data. But the really important thing to understand is that the noise is actually quite interesting also. So I'll be talking a bit about what the work that we've been doing with raccoons in North America, and and a bit about like kin structure, how it develops in natural populations, and also what the implications are for disease dynamics. So the population genetics part is kind of really underplayed. I usually very bad at going through methods and stuff like that because I find them quite boring. So so in case I've skipped over too much and you guys do have some questions about the methods that we're using, please feel free to stop me and I can explain further. So probably good to switch it on. Okay. So brief outline. Lots of people told me that you Guha, like you always do this. You just like jump straight into the talk. You never give this outline, and so nobody has any clue like what they're expecting or anything. So now I've started doing this, and it tells you like a brief path that we'll be taking. So I'm going to be talking a bit about what kin structure means, and also what are the demographic and landscape factors which are affecting kin structure in raccoons, and also how this impacts disease dynamics in these populations. Okay. And as you can see, raccoons are really cute. So skin structure is nothing but like the spatial aggregation of related individuals. And from a very simple demographic perspective, kin structure can develop when you have really low levels of gene flow in a fragmented population. So in this case, in this very extreme case, if there's no gene flow at all, each patch will just end up being one big family group, and you'll have really high levels of kin structure. However, when you start having a lot of movement between these patches, then this kind of kin structure gets diluted, and you get low levels of kin structure. So, but then how does habitat fragmentation and loss, which is one of the most important factors which is impacting species survival today, how does that really affect kin structure? So we all know what habitat fragmentation and loss is. I mean, it is the breaking up of this big patch into small isolated patches. And but increasingly we are starting to understand that we cannot just call it like habitat fragmentation and loss. There's been a lot of controversy on the terminology and everything. Earlier, just used to be called habitat fragmentation or just habitat loss. But now we're starting to understand that these are two very distinct processes. So there is habitat fragmentation, and then there's habitat loss. And these two can have very different effects on natural populations. So when we just talk about habitat fragmentation, if we have this constant patch size, we can just break it up into smaller and smaller patches. And in this case, what happens is that you have these patches which are low fragmented, and they have relatively low levels of isolation. You have these patches which have high levels of fragmentation and are really well isolated. So in this case, if you're looking at just movement of animals between these patches, what happens is that you have a lot of movement between these patches which have low levels of isolation, but relatively low levels of movement as the isolation increases. Okay, so what does that mean for kin structure? Well, obviously, as you have more and more gene flow, that kin structure is going to be diluted. And so here, you have low levels of kin structure, While here, because you have high levels of isolation, you're going to get higher and higher levels of kin structure. So the idea is that habitat fragmentation per se should increase the levels of kin structure at the scale of the patch. But what about habitat loss? So if you have habitat loss, what happens is that you get smaller patches. So in this case, habitat loss is not going to be affecting the movement of animals per se. It's only going to be mainly affecting the resources that are available to the animals in a particular patch. So when you have a big patch with a lot of resource, low levels of degradation, low levels of habitat loss, you're going to have high levels of resources, you're going to have high levels of birth rates, relatively low mortality, and you're going to have, and in the other one, you're going to, in the smaller patches, you're going to have low levels of birth rate, higher levels of mortality because low resource availability. So what you're going to be getting is this kind of like unidirectional movement from these higher patches into these lower patches. This is due to different mechanisms, but mainly like we've been thinking about it in terms of opportunistic phylopatry. So animals which are in these big patches, they're not likely to move until they're out-competed, and then they move to these relatively empty patches because they have low levels of birth rate, so there are some empty niches available. Okay? So, and then this could lead to source sink dynamics. Now, the main thing that we are focusing on are reproductive resources, right? So in the case of, and I wanted to mention this because I saw that there were some curious looks here. And in the case of raccoons, because that's the species that we're going to be mainly talking about, they are generalist mesocarnivores. They're not affected by like the availability of food or anything. And I'll be talking a bit more about this. They're mainly being negatively impacted by the availability of breeding resources. So in case there were some confusions there. So the idea is that if in this case, then you should be getting high levels of kin structure and in these high resource patches and low levels of kin structure in your small like patches which have high levels of habitat loss. Okay. However, when we look at the literature, there were, sorry, most of these things are gone, but between 2005 and 2011, Walker and there were two groups, Walkers and Banks, which like they were quite prolific, at least from an ecological perspective. And they published several papers, and they were saying that when you have a decrease in resource, and they were mainly talking about breeding resources, what you actually get is that you have high levels of kin structure in these low resource patches, and low levels of kin structure in the high resource patches. Their explanation was, one, it could be due to paucity of mates, so you don't have too much choice in who you mate with, and so you get higher levels of kin structure. Alternatively, they were saying that maybe it's because kin structure allows you to, or it encourages the sharing of resources. You're more likely to share these limited resources with somebody who's related to you, right, as compared to somebody who's not related to you. But we thought about the explanations, and we said that, well, you know, like cooperation and things like that are all fine, but they should be a product of something else, right? So we started thinking about it, and we thought that, well, one possible reason is that they're actually working with species that are really negatively affected by fragmentation also. So these are not species that are just dispersing very freely. And you also find that habitat loss and fragmentation are always correlated in natural landscapes. So when you have habitat loss, you will get, when you have low levels of habitat loss, you get these large patches which are relatively less isolated. However, when you have high levels of habitat loss, you're going to get these small patches which are more isolated, right? So the question is that like when these theoretical predictions do not match with empirical data, is it because of the fact that habitat loss and fragmentation are just correlated, right? And so what we're seeing is the signature of fragmentation per se, rather than the effects of habitat loss. So, and obviously, like species are affected by both isolation and resources. Now the question was, how can we test for just the effects of habitat loss? Well, obviously, the ideal thing is to just go out and say that, okay, fine, we take this nice pristine forest and we will just mow it down and we will make like nice patches, which are, in one case, we can control for just habitat loss and the level of fragmentation is exactly the same. On the other hand, we can control for habitat fragmentation and keep the level of habitat loss the same. But unfortunately, we did try and we were turned down. Well, no, we didn't try. But uh, in general, actually, there's a funny story with this because you know about the minimum critical patch size experiment of Lovejoy's in Brazil, right? So where they actually went out and they made these artificial patches. So, but the funny thing is that actually when I joined my PhD at Purdue, my prof was like, oh, like our first meeting, he was like, oh, well, we've been working in Peru, and so like, are you interested in going and working in Peru, right? So I was like, oh my God, like South America, brilliant, yeah, yeah. I was like, yeah, sure, like I would love to work in Peru. And so then he was like, yeah, we are working with raccoons. So I was like, oh, I didn't know the raccoons were there in South America. So I said, okay, whatever, what do I know about raccoons or South America or anything? But anyway, I was very excited about working in Peru. And so he said, well, do you want to go and visit the field site? So I said, yeah, I would love to go and visit the field site, right? He said, when do you want to go? I said, well, the sooner the better, right? So he said, what about this afternoon? So I was like, this afternoon, okay. I mean, like, I think I have to get some permission from the international students and scholars and, get some signatures and everything, but I'm ready. I mean, I don't need much. He's like, yeah, we can just drive there. So I was like, oh, okay, like, isn't it a bit of a long drive from Indiana all the way up to Peru? I mean, like, so he's like, no, it's Peru, Indiana. It's 2.5 hours from here. So my dreams of working in South America were unfortunately dashed, but we were working with a very cool organism, and that is the raccoon. And the raccoon is, I don't know how many of you even know about the raccoon, but the raccoon is kind of like it's a generalist mesocarnivore. It's gone to almost pest densities in many agricultural and urban landscapes in the US, right? And in general, we have this idea that raccoons are really successful in human-dominated landscapes, right? So, yeah. So the question is like, why are raccoons an ideal model species? To look at how habitat loss per se affects our level or patterns of kin structure. Okay, the first is that we do know from previous studies from our group that they are negatively affected by resource, and this is mainly breeding resources. So this is, I asked Jim, Jim, I'm going to be giving this presentation, and it'd be nice to have like a nice diagram. And he said, yeah, I don't have the time to send you the data or diagrams or anything like that. I'm really busy. So here you go, just take this paper. And so unfortunately, this is just a table. But these are the top 4 models that we are looking at the number of offspring, and these number of offspring are estimated by the number of placental scars on the uterus. And so we find that there is a significant positive effect of the number of dentries, which makes sense. So the more number of dentries, you have a higher number of offspring. And it's also affected by patch size, right? So the larger the patch, in general, you have more offspring. So this is a thing that raccoons are, or reproduction is affected by the availability of forest resources. Okay. The second is that in our previous paper, we had already shown that these raccoons are relatively unaffected by fragmentation. I want to quickly walk you through this graph. It's kind of so this is based on parentage analysis. So we are estimating dispersal distances using parentage. And so you find that, well, in general, within a patch, you have the highest proportion of assignments, which makes sense, right? So, and I also want you to note that, you can't really compare this with this in any logical way. Because the fact is that when we went out and trapped, we were trapping around like 85 to 90% of the raccoons in each patch. And this is from mark recapture data. So this estimate. So, but we only have 30 patches, right? And this is a landscape with like hundreds of patches. It's a 1,200 square kilometer area in the upper Wabash River Basin. And so we do not know. So yes, we can say that, okay, fine, raccoon A came from patch B, but if the raccoon came from a different patch which is unsampled, or it went to a patch which is unsampled, we do not have that information. So but we are expecting that the relative number of assignments should be proportional, right? So so if you just look at this, so basically there's one decision that raccoons have to make: do I leave my natal patch or not, right? And then there's a second decision that they make, that when I leave, how far do I disperse, right? In general, when you look at dispersal distances in animals, you will find like an exponential decay, right? So you have usually high levels of dispersal at the lower distances, and then it decays down as you go down. In raccoons, you'll see that there is this kind of like really nice normal distribution. These arrows are expectations based upon the number of number of patches which are separated by that distance class. So this is, if the dispersal is completely random, right, this is the level of dispersal we'll be expecting. So you can see that the dispersal, at least with respect to distance, is completely random. So it's not as if raccoons are more likely to go to a close patch as compared to a far away patch. It's just that they just move to a patch, right? And this is, so it's truly like Wright's like island model, you know, in some sense. Okay. The third probably important aspect is that obviously when you're looking at kin structure, you do want some variability in levels of kin structure so that you can test your hypothesis, right? So in raccoons, we get these patches which are what we are calling significant levels of kin structure. So this is based upon a randomization test done in Genelex, and it's based upon your related measure, which is Queller and Goodnight's relatedness. So you find that there are some patches which have significant levels of kin structure compared to random expectation, which is given by these gray bars, and some which are completely non-significant, which is just what you expect by random chance. So we classify these patches into two, like this SG patches and SN patches. And most of my talk is going to be talking about what are the differences between these two types of patches. Okay, so a quick thing about our study area. It's the upper Wabash River Basin, as I told you. It's highly agricultural, 80% or so. The patches, the forests themselves are about 15%. And 80% of these forests are like less than 5 hectares. So we're talking about a highly fragmented kind of a landscape, right? Okay. We have a total of 29 patches. We have more, but some of them are dropped because of low sample size. The number of years was less or something like that. We also have genetic data from about 1,067 raccoons. And we have serology from 500 odd raccoons. So the decrease by about 50% is, well, I mean, there were some raccoons that it was just, they started waking up before we could actually get the blood. There were some that, you know, like if you have a really long day in the field, the blood sample is, we don't have refrigerators when we're out in the field. So, and these raccoons are in the trap. So during the night, it's fine, but it starts getting hot pretty quickly. So you need to focus on the animals which could be in the trap. So you're trying to move as quickly as possible. So for multiple logistic reasons, we do have a decrease in our serology. Okay. But anyway, we do have about 556 raccoons, and these are all, these are the ones which we were able to get serological data for both canine distemper and these 4 serovars of leptospira. Okay, three serovars of leptospira. Okay. Okay, so what were our expectations as far as skin structure was concerned? Well, we expected that, you know, if you have high levels of phylopatry, that is the individuals which are born in a patch, stay in a patch, right? and you have low levels of immigration, right, we should be getting high levels of kin structure. So the SG patches should have high levels of phylopatry, low levels of kin structure. This site fidelity, unfortunately, once a raccoon is not, so we are trapping for multiple years. So we do know whether or not a raccoon is there in year 1 and whether it's still there in year 2, year 3, right? But if the raccoon is not there and is never trapped again in the subsequent 5 years, right, we do not know what happened to it. We do not know whether it died or whether it moved out, right? We can make guesses based upon the age of the raccoon, but we are just kind of clumping the effect of emigration and mortality into this. It's an inverse measure, so that's another thing that you need to remember. So we are calling it site fidelity, so it's a proportion of animals captured per year, recaptured per year. And this is basically 1 minus the probability that it emigrates or it dies, right? Okay. So we are expecting low levels of site. Oh, do I have it wrong here? Yes. So this is wrong. Okay. So we are expecting high levels of site fidelity in our this thing. So we are expecting low levels of emigration and mortality in the SG patches, and we are expecting high levels of emigration and mortality in our SN patches. So sorry about that. So this is the other way. Female sex ratio is another issue. Raccoons we've already shown are female philopatric, male dispersers. This is common in most mammalian species. So, so we did want to put in this female sex ratio because we would expect that if the sex ratio was higher, or if the female sex ratio is higher, we would have higher levels of kin structure. Okay. So as you can imagine, many of these variables are highly correlated, right? So because if you generally, if you have high levels of female FSR, you're also going to have higher levels of phylopatry. So they're kind of very correlated. So we didn't just directly put them into a logistic regression. Rather, we did a principal component analysis. So we got these 2 major components, which this thing for around, oh, this is also off. Sorry. This is the wrong way. So it should be 30 and 61. Okay. I did try to this thing. But anyway, the rest of the data is correct. Okay. So basically, we divided up into these 2 major components. And you will notice that the first component, the first principal component is basically what we are calling immigration phylopatry axis, right? So it's basically negative for immigration, and high levels are showing high levels of phylopatry. The second component is basically high levels of survival and female sex ratio. So the higher you are on this score, the higher your survival is, and your female sex ratio is also higher. And this is significant from a logistic regression perspective. But what I really want you to see is that there is this nice clumping, right? So basically, if you are in the SG patches, which are the greens, right, you have significant levels of kin structure. You're also likely to have higher levels of survival, higher levels of phylopatry, and similarly in the case of, or vice versa, right, in the other case. Okay. So this is exactly what we expected. So this is all going according to plan. And the second thing that we wanted to do was to look at how the landscape actually affects our levels of kin structure. So we have several landscape variables. So we again, they're all highly correlated. For example, agriculture, which makes up 80%, right, kind of dominates this entire thing. So if you have low levels of agriculture, you're going to have high levels of forests, and vice versa, right? Okay. So again, we did a PCA so that we could divide, or we could make them into these uncorrelated principal components, right? And we got several PCAs, but only one of them was actually significant. This is the principal component 1. And that you can see is basically a connectivity measure. So you have high levels of forest, high levels of connectivity. The patches are going to have high PCA scores, while if you have high levels of agriculture in the landscape, you're going to have a low PCA score. Okay? So So since there was only one axis, what we decided to do was I'm just showing it to you in terms of size. So basically, you have another thing. So the large patches or the large circles are these big, well-connected patches, and the small ones are these small, isolated patches. Okay, and in general, you find that high levels of kin structure are associated with these big, well-connected patches, which is quite opposite what we would expect in terms of other species, right? So usually, high levels of isolation have been expected to lead to kin structure. In the case of raccoons, because they are able to disperse across this hostile matrix, which is the agricultural fields, you find that it's actually your big, well-connected patches which have high levels of kin structure, right? Okay. So there is one interesting patch. So we are still like trying to get more dentry data, more fine scale this thing, because you will notice that there are some patches which are actually very small, very isolated, having high levels of kin structure, and vice versa. So compared to our demographic data, which is quite relatively clean from an ecological perspective, the landscape data is much more messy. And the reason that we are thinking that is that these raccoons may be actually being affected more. How much more time do I have? Like 15 minutes? Yeah, okay. By like very, very micro habitat kind of things, like dentries and water and things like that. So we are still trying to characterize these patches in more detail. Okay. Okay, so we do have kind of sourcing like dynamics, which is very interesting, especially from the perspective of raccoons, which are supposed to be very, very successful. So they're not expected to be negatively affected by these kind of factors. Okay. And as we expected, we're getting high levels of phylopatry, low levels of immigration in these SG patches, which are large and well connected. And also our levels of removal are low in our SG patches compared to the SM. So high levels of kin structure are kind of associated with these kind of characteristics. So the next thing that we wanted to ask was that how does this affect disease dynamics in these populations, right? So, well, when we talk about disease in natural populations, we're mainly looking at it in terms of contact rate. So if you have to transmit the disease, you need to come in contact, or you have to have some kind of a transmission between the infected and uninfected. But the problem is that when we look at contact rates, we can look at it at two very different scales. So at the individual scale, high levels of kin structure, and this has already been shown by other studies using contact collars, etc. High levels of kin structure, you're more likely to come in close contact with kin. For example, parent offspring. There's more likelihood of resource sharing. So contact rates are going to be higher when you have higher levels of kin structure at the individual scale. So this should be increasing our disease risk. So high levels of kin structure should increase our disease risk, right, from the individual perspective. However, when you think about it from a population perspective, I've already told you that low levels of gene flow, right, are going to be enhancing kin structure. So you actually have low levels of connectivity or contact between populations, right? And several modeling papers have shown that if you have low levels of connectivity in these fragmented populations, you actually have reduced disease transmission risk. So we started thinking about it, and we said, well, like we can't really publish a paper saying that, yeah, like we have no idea like how this is going to work. We have no predictions whatsoever. So what we thought about was that, well, the one thing that we can think about is in terms of the disease itself, right? So what is the scale that is most important for the disease? So if, for example, the disease is mainly being transmitted due to direct transmission, that is direct individual contact, and is mainly being affected by is mainly being affected by process at the individual scale, then we would expect an increase in disease risk. However, if the disease risk within a patch is relatively homogeneous, right, and you're mainly going to be affected by the contact rates between patches, okay? So we did have these two different types of disease. We had canine distemper, which was due to direct transmission. It needs like almost like nose-to-nose contact. So we expected, okay, fine. If this is the case, then we should increase our disease risk in canine distemper. However, leptospira is an environmental thing, so you basically infect a water body or something, soil, and therefore you transmit the disease. So if since raccoons, based on our radio collaring data, we know that raccoons move around quite randomly within a patch. So therefore, they should be like kind of homogeneously exposed to the disease within a patch. And in the case of leptospira, therefore, kin structure should actually reduce our disease risk. So these are just our expectations. So the first thing that we needed to check was, is this question of scale actually relevant, right? So what we did was we used a measure called co-exposure. So basically, it's kind of like you take two random individuals, you just ask the question, are these two both infected, right? And then you can calculate a probability that two individuals are both infected with the same disease, okay? Relatively crude measure, but that's the only thing that we had. So what did we ask? So first thing we asked was, how does kinship affect our disease transmission risk? And very much as we expected, in the case of canine distemper, half-sibs, so we decided half-sib or higher was our criteria, because these are the groups which are going to be more in contact with each other, okay? So half-sibs are more likely to be co-exposed as compared to unrelated individuals. In the case of the leptospira, there is no significant difference between our half-sibs and unrelated. So basically, as we expected, this kinship is not really affecting co-exposure rates in leptospira because it's kind of an environmentally transmitted disease. You're just as likely to be exposed whether or not you're a half-sib or you're unrelated. Okay. Okay, so the next thing was contact rates between patches, right? So we used our estimate of gene flow, which was the effective number of migrants between the patches using the genetics. And what we found was that in the case of leptospira, at least in two of them, there was a significant positive correlation. This is based on a Mantel test, and this controls is a partial Mantel. So it's controlling for the distance between the patches, right? And we're looking at co-exposure rates between individuals in two different patches. So the probability that individuals in two different patches are exposed to the same pathogen. So we are finding that disease transmission or co-exposure rates are positively correlated, right, with gene flow in the case of the leptospira, but it's negatively correlated with gene flow in the case of canine distemper. So basically, because, as I mentioned before, kin structure, right, is actually negatively affected by gene flow. So no, negatively affected. Okay, negatively affected by gene flow. So so basically, you're in the case of canine distemper, you actually have a negative relationship. So the more the gene flow, right, you have a lower risk of being co-exposed in the case of canine distemper. And that, we don't have a real explanation for it, but one possibility is that when you have a lot of gene flow, right, raccoons are solitary animals, they're not social, right? And so when you have a lot of gene flow, you have all these non-related individuals coming into the patch, and they really don't have really close contact with each other, right, except maybe during mating season or something like that. So this may be the reason. Okay. So we definitely know that, okay, fine, our assumption that the scale of contact this thing is definitely okay. So canine distemper is definitely being affected by contact rates at the individual scale, while leptospira is being affected by contact rates at the population scale. Okay, so now how does that actually affect disease exposure? So our index of kin structure, in this case, we actually had to go to a kind of a continuous variable. So we used, because of the differences in sample size between our populations. So what we did was that we classified these individuals into related or unrelated using ML relate. And so basically, related means they were half-sib, related as half-sib or higher. And the reason that we're choosing half-sib, I want to reiterate, is that these half-sibs or higher are the ones which are likely to be like offspring of the same mother. They're more likely to be in contact with each other. So from a disease perspective, this is the level of relatedness that we're really interested in. Okay. And we calculated for each patch, we calculated the proportion of individuals which were half SIB or higher. Okay? And our index of disease exposure was just the seropositivity. We used generalized estimating equations. Our dependent variable was a binomial variable, so it's the number of seropositives given the number of individuals which are tested. And our index, our main dependent or independent variable was this index of kin structure. We also control for landscape factors because we already know that kin structure is being affected by landscape factors. I'm not talking about that right now. So what did we find? Well, as we expected, high levels of kin structure increased disease exposure risk in canine distemper. So for a 4%, there's a 4% increase in disease in seroprevalence rates for a 1% increase in our index of kin structure, which is the percentage of individuals or proportion of individuals which are related half SIB or higher. Okay? And in the case of Leptospira, we actually have a decrease. So higher levels of kin structure are actually decreasing our disease exposure risk. And this is between 1.5 and 3% decrease for every 1% increase in kin structure. Okay. So just to quickly recap, so we have when the transmission scale is at the individual, that is in the case of these directly transmitted diseases, kin structure actually increases our disease exposure risk. In the case when the transmission scale is the population, for example, in environmentally transmitted diseases, our kin structure is actually decreasing disease exposure risk. Okay? Okay. So I quickly want to like make some concluding statements, and that is, well, the first thing is that looks can be deceiving. Raccoons are considered to be really successful in these fragmented landscapes, right? They're almost a pest. They've reached a pest species status. But what we're finding, right, is that raccoons are also negatively affected by habitat fragmentation. So it is not that raccoons are doing uniformly well in this fragmented landscape. It is just that they're able to disperse to patches which are actually not supporting reproduction and therefore maintaining a high density across the entire patch. So for example, if raccoons were not able to disperse to these patches, we would have more and more empty patches because they're not able to support a self-sustaining population, and raccoons would probably be also endangered, right? So it is just the fact that they're able to disperse rather than the fact that they're unaffected by fragmentation, which is important. This is, I have a personal bone to this thing. So it took us 4 years to publish this paper, and it finally went into animal conservation. And so I am very grateful to the editor of Animal Conservation, so I just wanted to mention that. But 4 years for us to convince people that raccoons are also negatively affected by habitat fragmentation. And this is with like a lot of genetic data, a lot of GIS, a lot of stuff. And it took us a lot of convincing to, so for the students, you know, perseverance, and eventually, like, you will get it published. Okay. The second is isolation by resource. So we've all heard about isolation by resistance. We've heard about isolation by distance, right? But what we are finding is that there is this completely different kind of functional isolation, which we are calling isolation by resource. So because in a fragmented landscape, you have very heterogeneous levels of resources, individuals are less likely to disperse from high resource areas, right? So when we look at animals or species which are affected by both fragmentation and loss, right, there's going to be this double whammy. So one is that you're going to be having high levels of genetic differentiation simply because the individuals in these isolated patches are unable to disperse from them, right, because of the large level of inhospitable matrix between that patch and another patch. The second issue is that individuals in big, well-connected patches are unlikely to want to disperse, right? So we have two different processes which are going to be driving genetic differentiation and therefore genetic drift and all these processes in these fragmented populations. The third is that, well, all diseases are not equal. So we cannot say that, okay, fine, we've got this prediction for one disease. We need to understand the biology of the diseases and how it's going to be affecting the transmission dynamics and therefore our predictions for that disease. So we have some really, we are hoping for some really interesting this thing, because we've gone out and we've actually done a large-scale experiment removal of raccoons in, I think it's 20 and 20, 40 patches across this landscape. These are not these patches, but they're a different set of patches because these are our long-term patches. But so we have the serological data from before the removal. We have the serological data from after the removal. And now we've been following these removed patches for the past 3 years. So it's going to be really interesting to see how, when we remove animals from a patch, right, how that's going to be affecting disease dynamics. And we have certain predictions, and I'll be happy to talk about it later. But I think that the important thing to understand is that one major control mechanism for diseases in natural populations is to go out and cull these populations which are infected, so that you do not just have a wildfire spread. And so I think that what our data is going to be saying is that, okay, fine, like when we do that, how is that going to be actually affecting disease dynamics? Okay. So quickly, large number of landowners who have really like opened their hearts and their land to us to go in and trap raccoons. Many of them were like, "You're not removing them?" Like so no, like we actually trapped them and released them back, and they were kind of like irritated with that whole thing, because raccoons are a major pest. But and India is a wonderful place. I mean, like the people are really warm and really hospitable. And the really interesting thing about farmers in Indiana is that they really have no concept of distance, right? So Jim is from New York. And so they were like, "So where are you from? You talk strange, right?" So Jim is like, "I'm from New York." "Oh my God, you're from New York?" And then so I would be like, "I'm from India." And it's the same expression, same tone. "Oh, you're from India?" So if you're not from Indiana, you're from far, far away. That's their concept. And so it's really wonderful to work with them. Obviously, all the members of the Rhodes Lab are field and lab technicians, and this funding was provided by Purdue. And I'll be happy to take questions, and I hope I'm on time. Yes? Okay. Well, co-exposure. So basically, if you take two individuals and you ask the question, are these both seropositive for canine distemper, right? That is negatively correlated with gene flow. Yes. Correct. Right. So this is a very good question, right? And I think that the reason that it gets complicated is the fact that gene flow is negatively correlated with kin structure. So the more gene flow you have, right, the less kin structure you're going to be getting. Okay. So when you have high levels of gene flow, yes, you're going to be getting more likelihood of contact between these two populations, but the kin structure within each population is being diluted. Okay. Two individuals which are unrelated, at least in raccoons, they're not likely to be coming in really close contact with each other, right? This is not our data. This is basically contact collar data from other people. So the idea is that when you have these unrelated individuals, even if they're like moving freely between the patches, right, you're not going to be getting very, very high levels of co-exposure. Now, the increase in co-exposure due to reduced gene flow, I am thinking it is more because when you have high levels of gene flow, so it's kind of like a artifact, and that I should have pointed out. And the artifact is coming out because we are just having a Boolean variable, right? Infected, non-infected, right? When you have low levels of gene flow, you have high levels of disease exposure within that patch, and so you're just more likely to get two individuals which are positive for canine distemper. So it is a good point, but this is the best variable that we had in these natural populations. Now, I guess if you actually go out, maybe doing some kind of sequencing work or something like that, we'll be able to get maybe more fine-scale thing, but we haven't really thought about it. That's a very good question.